Epilepsy is often thought of as an electrical storm in the brain, sudden, uncontrolled bursts of neuronal firing that disrupt normal function. But underneath the surface of this hyperexcitability lies a complex metabolic landscape, one where the balance of energy supply and demand is constantly in flux. For years, researchers have focused on neurotransmitters, ion channels, and genetic mutations in the search for causes and treatments. Yet one of the most overlooked contributors to seizure activity may be a far more fundamental player: cellular energy. Specifically, brain glycogen, the astrocyte-stored reserve of glucose, may play a crucial and underappreciated role in modulating seizure risk, severity, and recovery. Glycogen in the brain is not widespread. It is stored almost exclusively in astrocytes, which act as support cells to neurons. Under normal conditions, glycogen is broken down into glucose, 6-phosphate or lactate, and shuttled to neurons during periods of intense synaptic activity. This astrocyte neuron energy exchange helps maintain neurotransmitter cycling, buffer excitatory signals, and keep ion pumps running, critical functions during high-demand periods. In epilepsy, where neurons fire excessively and unpredictably, energy demands skyrocket. Glycogen, in theory, should be mobilized to buffer against these surges. But this system is far from foolproof. In epileptic brains, especially those chronically exposed to seizures, glycogen metabolism often becomes dysfunctional. Some studies have shown that glycogen content is abnormally low in epileptic tissue, suggesting chronic depletion. Others have found excessive accumulation of malformed glycogen structures, such as polyglucosan bodies, which are poorly degradable and may interfere with normal astrocytic function. Either scenario, too little usable glycogen or too much unusable glycogen, can impair the brain's ability to meet the sudden energy demands of a seizure or recover afterward. This metabolic vulnerability may not just be a consequence of seizures, it could be a trigger. Seizures are energy-intensive events, requiring massive ADP output to support ion channel activity, neurotransmitter release, and synaptic reorganization. If astrocytes cannot rapidly supply lactate and glucose through glycogenolysis, neurons may become energy-starved mid-firing. This can lead to loss of membrane potential control, accumulation of extracellular glutamate, and runaway excitation, conditions ripe for seizure initiation or prolongation. In this light, energy mismanagement is not peripheral to epilepsy, it is central. There is also evidence that impaired glycogen turnover disrupts the balance between excitatory and inhibitory neurotransmission. For example, synthesis of GABA, the brain's primary inhibitory neurotransmitter, requires both glutamate and energy. If astrocytic glycogen stores are dysfunctional, GABAergic neurons may be unable to maintain inhibitory tone, tipping circuits toward excitation. This may explain why some forms of epilepsy are resistant to drugs that increase GABA activity. The underlying energy deficit remains unaddressed. Interestingly, this energy-centric view helps explain why ketogenic diets are effective for many patients with drug-resistant epilepsy. By dramatically reducing glucose intake and forcing the brain to rely on ketones, these diets reduce the metabolic volatility of glucose-based energy systems, stabilize neuron firing, and minimize the need for glycogen buffering. In essence, ketogenic therapy bypasses a broken system, offering an alternative fuel that's both more stable and less dependent on astrocyte neuron glycogen exchange. This insight also opens the door to new research directions. If glycogen mismanagement contributes to seizures, then stabilizing glycogen metabolism may reduce seizure risk or severity. This could involve modulating glycogen synthase or phosphorylase activity, enhancing astrocyte neuron metabolic communication, or developing therapies that prevent the formation of non-functional glycogen aggregates. Targeted antioxidants or mitochondrial support compounds might further help astrocytes maintain energy balance during high-stress events. Yet this area remains underexplored. The standard clinical approach to epilepsy rarely considers the brain's energy economy, focusing instead on electrical patterns and synaptic signaling. But as we learn more about the metabolic dimensions of neurological disorders, it becomes clear that energy management is a prerequisite for electrical stability. Without it, the brain becomes increasingly fragile, and the line between normal function and seizure becomes dangerously thin. In conclusion, glycogen may be more than just a backup fuel. It may be a metabolic gatekeeper of seizure control. When well-regulated, it allows the brain to absorb energy shocks and recover quickly. But when mismanaged, It leaves neurons vulnerable to collapse under the weight of their own activity. Recognizing and targeting this vulnerability may offer a new frontier in the treatment of epilepsy, one that doesn't just dampen electrical noise, but stabilizes the energy foundation upon which healthy brain function depends.